All right, I guess I have my marching orders. Amy says it's time to get started. Everybody has their popcorn. For those of you who don't know me, my name is Karen Churchill, and I'm the President and CEO of AgWest Bio. And welcome. Thank you so much. I certainly appreciate to see some familiar faces and coming out in the snow. So it's great to see everyone here tonight. And of course, we like to start our events by acknowledging that we are on Treaty 6 territory and homeland of the Métis, and recognize the role that the First Nations and Métis played in reestablishing and reaffirming our reconciliation and collaboration with each other. So we're very pleased tonight that Dr. Kevin Folta is joining us from Florida, braving our winters, if not willing to brave a Florida hurricane. We can understand that, I think. And just a few housekeeping items. So please remember to turn off your cell phone or switch it to vibrate. And we encourage you to tweet, put whatever out on social media using the hashtag ABIC Speaker. And there'll be some time for questions afterwards, of course. And we have a survey. And participants, of course, have a chance to win an AgWest Bio gift box. How could you not do a survey for that, right? And if those of you who don't know very much about AgWest Bio, I think everybody here does, but AgWest Bio is the Bioscience Industry Association. We're a catalyst and a connector of research in bioscience, and we work to develop bioscience-based businesses in the province and grow the bioscience. So our members include a wide range from public research institutes to private companies, both small and large, economic development agencies, government agencies, and private individuals, some like yourselves. So we always encourage you to join AgWest and help us with our causes. And a little bit about ABIC. I actually always have to look up. ABIC is the Agricultural Bioscience Innovation Center. And we rarely ever say anything other than ABIC. And it is a foundation that is a charity that is dedicated to promoting science as a powerful tool for solving the world's and global challenges. And this speaker series in particular allows us to bring in incredible speakers from around the world who will talk to us and inspire us and help us learn about the bioscience by sharing their bioscience industry, by sharing their stories. And I have to give a special shout out to Brent Zettel of Zias, because Brent has been consistently being an incredibly generous benefactor to the foundation and makes these events possible. So this past year, he donated $300,000 to the ABIC Foundation, and we'll use that money to grow biosciences and help train entrepreneurs. So he's also very dedicated to the bioscience cause. And now I should also get to introducing our guest speaker. So Kevin is a professor of the Horticultural Science Department at the University of Florida, and his program focuses on the role of light in plant development and the genomics of fruit traits. So his family has a mixed farm, not like we would think of a mixed farm here in Saskatchewan. He grows fruit, vegetables, eggs, and turkeys. He has been recognized with a very prestigious Borlaug Award in the Agriculture Communications. And many of you, including myself, probably follow his podcast. So I didn't realize there was two. I follow the Science, Facts, and Fallacies podcast, but there's also Talking Biotech podcast. So please download them. They're very interesting. And my last little tidbit is he's actually a city kid from Chicago. So he's come a long way. He's seen the light. And with that, please, I'd like to welcome you to the stage.
Wow, well, thank you very much for that. That's very nice. It's wonderful to be back in Saskatoon. It seems like I always end up in Saskatoon for some reason, every time I come to Canada. It's an interesting place that always kind of land here. Well, thank you very much for coming out tonight. I appreciate it. I know the weather sometimes this is a beautiful night, right, for Saskatoon? This would be the coldest night ever where I'm from. But the reason I'm so excited to be here and why I was so happy to get this invitation is because we hear too much doom and gloom about science. And every time we turn on the news channel or watch a science fiction film, it's about where science goes bad, where science goes wrong, or someone trying to scare us about new innovations. But as a scientist who's studied science his entire life, and I'll show you pictures to prove it, that I've been in awe at what's happening right now, what's on the edge of coming next. The fact that people who suffer from some of our most profound illnesses will soon find therapies and cures. In other cases, we'll see new ways to feed people around the world more nutritious crops. And to me, through my lens, I see so many good things coming that I really wanted to share them with you tonight. So we'll take a little cross-section, across a walk of biotechnology and some of the big things that are happening. The first thing that I'll show you, though, is we'll start out with really where my career in science started. And many of you in this room might remember what happened in July of 1969. I was 2 years old, but I can remember watching the lunar lander on a small black and white TV in Chicago. We lived in an apartment upstairs from my grandparents. And my father instilled in me that this was something I needed to remember. And that was really cool. And then not long after that, I was 4 years old, and was watching this guy, Mr. Rogers. Mr. Rogers had a song called "You Can Never Go Down the Drain." And in that song, he had a line that was, "You're bigger than the water." Bigger than the water? I mean, we got glass of water, we got ocean. What do you mean about bigger than the water? And I asked my dad, bigger than the water. And he introduced me to the idea, of molecules, of atoms. He took a gross pen out of his pocket. He's worked in engineering and electronics, and always had a pocket full of screwdrivers and tweezers and things. And he took out a dirty, gross pen and popped that and showed me a little purple tip on there and said, "There are more molecules in the tip of that pen than you can even count or imagine." So we were talking about planets, we were talking about molecules from time very early. And I was super psyched because I was going to get to go to kindergarten in a year or two. And you know what school meant? Science fair, right? So I'm totally on fire about science fair. I'm walking around the house all the time, science fair, science fair, science fair. My mom's like, "Will you shut up about science fair?" I was on fire about science fair. I got to grade school and learned that science fair was only for kids between 5th and 8th grade. I was shattered. When you're in kindergarten and 5 years old, getting to 10 is like a lifetime away, right? I had no urge to go on at that point. I complained enough that Mrs. Mutter decided to pull some strings and was able to get me into the science fair. And I presented the charismatic megafauna of the Cretaceous, you can't really see me there, of the Cretaceous tertiary boundary. I was the first kindergartner ever in science fair. And not long after that, I pretty much was on my way to being a professor, and my parents dressed me that way. That's me. You can't really see the striped pants, but I had a big armful of books and bowl haircut, and I was on my way to being a university professor. So all of that happened. But what were the things that were really most important to me and my family growing up in an apartment in Chicago? We had lots of friends who were in rural areas, and we knew how important farms were to our economy as a nation and also to feed people. We were very excited still about talking about solutions for the developing world. We talked all the time about people who had very little, especially not enough to eat. We talked about people making better decisions in terms of food choices and eating the right stuff, even in the industrialized world where we had plenty of food. And even with all of that, we talked about the environment. We spent a lot of time out fishing and exploring nature. And it was a major reason for me staying in science, was because of all of these reasons. How do we think about food and fairness around food and equity among food? That people would have the opportunity to have access to food and do this with environmental sustainability. And these were values I grew up with from very young. It transitioned really well into my university career as we studied as we were the first laboratory to sequence the strawberry genome in 2010. So we had a blueprint of all the genes and all the genetic controls that made a strawberry a strawberry. And it was a very interesting project that we did by financing it by passing the hat and twisting arms, whereas all the other plants that were sequenced had big budgets. We were able to do it on a shoestring. It was really cool in that regard. And I also do a lot of work with controlled environment agriculture, understanding how we can grow plants in novel environments just by playing with light in order to get them to do what we want them to do. Plant whispering, the idea that we can tell a plant how to grow by speaking in a language of light. And we play with interesting ways to kill plants, coming up with new molecules that can help us combat noxious weeds that are safer for the environment and safe for us. So these are the things that I do today. There are a lot of folks that, then one of the things that has happened after, during my time as a professor was I got married to a farmer. And she was originally from Ukraine, grew up on a farm. She's sturdy and lifts 2 50-pound bags of feed without a problem. And she really taught me a lot more about putting what I'm learning in the laboratory into action on the farm. And it was super exciting because we sell at a farmer's market every Saturday morning. We always have great stuff there. We raise turkeys. We grow lots of really interesting fruit. We have a lot of interesting animals on the farm. And we have one more interesting farm animal we're adding in a couple of months, which is really cool. Yeah, so my favorite breeding project. So, yeah, so it's a really great time, and I'm so excited about teaching my daughter about technology. As soon as she gets here, she'll be one day going science fair, I hope. Hope she inherits that gene. The bottom line is I think technology is totally cool, and I am so excited about the things that are happening in agriculture right now, and in medicine, and in conservation for that matter. There's new ways of growing plants in funny environments to keep plants growing near urban centers, employing local people and cutting carbon footprint. It's exciting to see what's happening on the large farms, where GPS is driving tractors, where drones and sensors are allowing farmers to farm with fewer inputs and lower costs. We're also seeing drones playing a role in doing things like mapping fields and learning where pathogens are, so sprays are limited. And in medicine, the advent of robotics and human surgery, the new imaging techniques, and the new therapies that are coming along are beyond imagination. Things that we wouldn't even have imagined 10 years ago are happening now in hospitals. Probably the biggest breakthrough happens at the level of DNA. Can we, in fact, manipulate the molecules of life themselves in order to find beneficial therapies? And that's what we'll spend most of our time on today. Nonetheless, there's still people who are not as excited about technology, and some people who feel that the old way of doing things is still better. And I get that. Maybe there's something kind of interesting and traditional, or maybe something kind of folksy about, digging holes using it with a hoe rather than a tractor, or, sawing off a limb with a saw without anesthesia. Maybe even some of the diseases we've left behind because of breakthroughs in medicine. There's still a lot of folks who feel that way, and some people who are very uncomfortable about the newest technologies, whether they're in plants or whether they're being used in medicine. And I understand where they're coming from. New technology can be scary. It can, especially when you don't understand it. That's why tonight is so important, is because I would like to show you some of the really hopeful, exciting stuff that's coming. And at the end, I'd be happy to answer your questions. And I'll stick around till I got a 5 a.m. flight tomorrow morning, so I'll stick around till we're done. So why is there a difference? This is the big question. Why is there something different between what we know as a scientific consensus and what the public believes? And this is really an important question for the time. You don't have to look further than the COVID pandemic to see how bad information and bad communication can really muddy the water of science. Whether it's people online saying there's 5G microchips in the vaccine, to our esteemed national leaders in our country and across North America, who are fumbling with communication strategies and not communicating to us in clear ways that make us comfortable with new technology. And this is one of the major problems and one of the major reasons why there has to even be controversy. So how do we fix it? And it's by having events like this. So thank you very much for having this. This is pretty exciting stuff. So let's talk about what this is and what this isn't. What is biotechnology and what isn't biotechnology? We'll talk about its applications in agriculture. We'll talk about applications in medicine. And at the end, we'll talk about what does the future look like? What are some of the things we can anticipate coming next? The best part about that is I really have no clue. The best part is talking to my classes every year and telling the students in molecular biology that the job you will take, the things you will invent, the innovations you will enjoy have not even been invented yet. And that's what's so cool. The major technologies we're using today didn't exist in 2013. And this is why these fields are so exciting and why we're paused for such tremendous breakthroughs. On top of that, there's been great investment in the areas of biotechnology, primarily in other countries, but somewhat in North America as well. So let's start at the beginning. If we're talking about biotechnology, we have to talk about where it starts, and it starts in DNA. And what is this stuff? Well, DNA is the blueprint of life. The DNA in any one of your cells is the blueprint of you. It describes all of the different, it contains the information to make all of the proteins, all of the parts that put together your cells. And whether it's in your toenail or on the top of your head, it has the same DNA in all those cells. So that's what's really cool. It's a master blueprint that's put away in all of your cells. This is one I really wanted to show you here, is that any one single cell contains a number of chromosomes. And if you were to unwind one of those chromosomes, don't try it. It's really hard to put back together. You have DNA, and DNA is represented by this double helical molecule. It's very familiar to everybody, and has 4 different chemicals that it's made up of. This stuff called adenine, guanine, cytosine, and thymine, we usually talk about as A, G, C, and T by their initials. It's the order of A's, G's, C's, and T's that makes up a code that your body knows how to decipher, to turn into the proteins that do the job as enzymes, to make metabolism run, to make the structures that make your cells strong. This is where this information all comes from what's present in DNA. So think of it as a hard drive. There's information on the hard drive of your computer tucked away deep inside the back of a computer. Hopefully you never see it. Hopefully it's put away and always with great integrity. It accumulates some errors over time. And in that way, it's a lot like the DNA in your cell. The DNA is a permanent storage, put away version on a hard drive. There's another molecule we call RNA, a lot like the USB drive. It's temporary. It contains only a tiny bit of the information that's present on the hard drive at any one time. It's subject to degradation, especially if you live at my house. I pull one out of the dryer every week. Ultimately, you can take that information on the hard drive to work where they have a good color printer and print out the information you need. Think of that as the proteins of the cell. So what I've really outlined here is what's called the central dogma of molecular biology. Information that's put away in one place in a safe, secure way, that a little piece of this information can move in a temporary form and then ultimately go to a place where it can be used. That's what they refer to as the central dogma. DNA in the center of the cell, this RNA intermediate, and then ultimately a thing called a ribosome is like the cell's printer. It's where the RNA message and information goes to be translated into the proteins, the thing that comes out the end, like the print that comes out of a printer. It's the product of the cell. What's really cool about that is that it's a really nice analogy. It fits terribly well. So today what we'll talk about really is how do we put more information on the hard drive or take information off? What happens if we can put some RNA into a cell without it being on the hard drive? We'll talk about all these things here today. So how do we change the information on that hard drive? Well, let's talk about libraries. Libraries are tremendous collections of information. And so if we want to change the information in a library, how do we do it? Well, we can put a new book on the shelf. That's one way to do it. If you're from Florida, you can take a book off the shelf and light it on fire. So these are two ways that we can do. You put information in, you can take information out. But today, there's new technology, by analogy, that allows us to go into the library, into a library, find a book, a page, and eliminate one word to substantially change the meaning of the sentence. Maybe we take not out of thou shalt not covet your neighbor's stuff. Now game on, party time, right? So this is the idea that you can either add information to the library, you can take information out, or you can change one piece very precisely to change its meaning 100%. And this is what we're doing in biotechnology, only we're using the library of the cell's information. We can add a new gene, new information to make something new. We can eliminate a gene to stop making something that was previously made. We can go into a single gene and erase a letter or two, or change a letter or two to correct it or to eliminate its function. I'll show you examples of all of this today, and this is really the analogy I wanted to work from. The development of the SARS-CoV-2 vaccine. This was an amazing breakthrough in biotechnology in that it allowed us to use a technology that's been in development for some time with a target of public health. And the idea here was to use the cell's USB drive, the RNA, and now inject that RNA into a muscle cell, let it go on to the cell's printer, the ribosome, and create the product that ultimately would confer immunity. And it worked by taking taking the cell's USB drive and having that produce the spike protein of the SARS-CoV-2 virus. That particular protein then would be secreted by the cell to induce an immune response. That mimicked the coronavirus. So it was a way of telling the cell, telling the body that the virus is there even when the virus isn't. Because this protein that's a signature on the outside of that virus has now been created. So this is a great way to kick off innovations in biotechnology. This didn't exist 4 years ago. Well, 3 years ago it didn't, just barely. And so this is what's so exciting about this particular technology, is its agility, its ability to change and be used to meet the newest public health threats. But let's talk about the beginning of the biotech explosion. Where did this happen first? And this is so cool. This goes back to the 1970s when the first major breakthroughs in biotechnology began. It happened because of diabetes. Diabetes is a continual problem that's increasing in society, and today some 43 million people are taking insulin in order to control diabetes. It's a complication in other diseases and can lead to other problems. So it's important that it's controlled. And it's controlled with administering insulin, a naturally occurring hormone that is typically deficient in diabetics. In the old days, we used to make insulin for diabetics by going to the slaughterhouse and starting out with 10,000 pounds of pancreases. 10,000 pounds of pancreases to create 1 pound or half a kilogram, I forget I'm in Canada, half a kilogram of insulin. So an amazing distillation from this source. And Eli Lilly was at the forefront of this. These are some really cool old historical photos. There's the Eli Lilly and Company gut bucket with someone sorting pancreases into a grinder in order to make human insulin. So this isn't such a bad way to do it. Problem is, a lot of other contaminants come along. Other things that contaminate, that could cause allergies or other problems that were coming from the animals that they would process. So what if we could take the gene from a human being and put that into a bacterium and let the bacterium make 100% pure human insulin? And that's exactly what happened next. The human gene was cloned out of a human cell, placed into a bacteria, and in a giant fermenter, you can make massive amounts of insulin that's 100% pure, 100% human. And this is why this was such a breakthrough. It was insulin, it was growth hormone. When I was a kid, I was learning this stuff. Growth hormone that would help people with growth deficiencies, long bone growth deficiencies, who had short stature in life, that they would get one treatment of growth hormone by purifying the pituitary glands of 50 cadavers. You now could do it in bacteria. These were huge breakthroughs in biotech. Recombinant DNA technology, that when you see a vial of humulin, R, it means our DNA, recombinant DNA, that DNA was cut and pasted to new places. And this is the basis of how this product works. So how do we do other tricks with genetic engineering? Genetic engineering of plants is a pretty popular topic. And most people don't understand the black box behind it. And I'd like to make it horribly simple for you to understand. It's a really pretty cool process. So have you ever taken a basil plant, or what else really does this well? Basil does it well, and other flowers. You cut one off, stick that cutting in water, and it grows roots. The cells on the bottom of that cut surface understand that they are no longer connected to roots, so we better make some. This is a cool process that plants can do that animals can't. For the most part, regenerate the parts that are missing. It's pretty cool how this works. And so can we exploit this property of plant cells to be able to take a single cell and have it grow into a whole new plant? And this is a process that was really explored back in the 1940s and 50s, that they would take carrot cells and then be able to take just one cell or a couple cells, put it into the right media, under the right petri dish of stuff, right nutrients and right hormones, and then could create a brand new plant from that one single cell. Pretty cool. Now the trick is, and this is what it looks like in real life, you take that one cell and it eventually starts to differentiate and divide and make new leaves and makes a whole new plant. Really, really neat topic or process. And you can do this with many kinds of plants. So what if you could, this is the way the internet says you do it. Not quite right. The way it's actually done is we use Mother Nature's genetic engineer. This is a bacterium called Agrobacterium tumefaciens. It's found everywhere in the soil. And what it does is it lives its life cycle by infecting a plant. It takes a little piece of its DNA and installs it in the plant cell, and the plant starts to grow really quickly with this mass of cells they call callus. This is set up so that the agrobacterium can live there and be happy. It's like the roommate that comes over and throws in its suitcase, rearranges your house, and says, "I'm staying." That's what agrobacterium does. And scientists studied agrobacterium and said, instead of it delivering what it wants, what if we can get it to deliver what we want? That is when plant biotechnology was born. Because we figured out that if you could get your favorite gene in that one cell, and then turn that one cell into a whole plant, now that whole plant would carry your favorite gene. And this was worked out by several independent groups in the 1980s, 1983 to be exact, and became the beginning of plant biotechnology. So how do you use this? And probably one of the best applications has been developing plants that can defend themselves. You know, we've all seen the tractor image spraying the field with pesticides or trying to control some sort of fungus or insect. And controlling insects is really difficult and increasingly a challenge. The big problem here is that the insecticides cost money, it takes labor, it takes fuel to deploy them, and nobody wants to put extra pesticides into the environment. So what if you could get a plant that would produce something to protect it? Well, plants already do that. They produce lots of compounds to protect themselves. But what if you could come up with a specific one and give the plant the genetic instructions to produce it? And there's a bacterium called Bacillus thuringiensis, or BT. And Bacillus thuringiensis, when it's consumed by an insect, kills the insect. It's very specific towards certain worms and beetles, very selective. It doesn't work well on other insects, just a very narrow range. It's so popular that it works so well that it's used as an organic farming, organic gardening spray. BT, it's natural, so you can use it in organic farming. Works really great. The problem is that if you're an organic farmer and you apply it and it rains or the sun comes out, it washes off. So people started to think, what if we could take the gene from Bacillus thuringiensis that makes this little toxic protein, and instead of spraying it on, have the plant make it? And that was the idea. Could you have a plant, instead of a caterpillar coming to eat the plant, You would have the Bacillus thuringiensis, little piece of DNA, and use a recombinant DNA technology to introduce it to the plant, that now it would kill the insect. You would not need to spray any kind of insecticide. It worked brilliantly. And the best part is the results in suppressing the amount of insecticides that needed to be applied. On corn specifically, it was cut dramatically over the time that it was originally used. This only goes up to 2011. I had trouble finding one more recent, but it gives you an idea of the insecticide in terms of kilograms of active ingredient per hectare really came down because BT allowed the plant to protect itself. It really worked well and helped improve the quality of corn. It's interesting that in Florida we produce a lot of sweet corn, and almost all of it goes to the EU. We don't use any BT sweet corn. They don't like the GMO corn, or the genetically engineered corn. And what that means is the spray planes come over and spray an insecticide almost every couple hours to protect that crop. So here's a case where by not being able to send it to the EU because of the genetics, we now have to rely on more chemicals, more fuel, and more carbon generated. It seems like we're going backwards. So the best application of BT may have been in the eggplant, or the BT brinjal, as they call it in Bangladesh. It was originally designed for India, but the Indian parliament says we don't want the genetically engineered plant, the engineered crop. And the eggplant's very important in India as it is in Bangladesh. The parliament in India says we decided we don't want it, it's been approved, but we're not going to accept it. The ag minister for Bangladesh says, I will take it. She's 4 feet 11, she's excited about technology, and she said, I want this for my farmers and for my environment. It will allow us to use less. That farmers there are still using old school insecticides, things that have been outlawed here for ages. And they're putting them on with a scarf over their neck, over their face, usually with no gloves and no protective equipment, spraying out of those old school sprayers. As they have an acre or two of plants to protect, this is their livelihood, they use whatever they can to protect it. And their kids are out there spraying chemicals too. That ended with the introduction of the BT eggplant. The BT eggplant allows subsistence farmers in Bangladesh to produce beautiful fruits with no insecticide treatment. This went from a few farms in 2014 to now thousands of farms, and they're even smuggling those seeds into India. Because you can't stop good technology from the people who need it. People want innovation. So let's talk about some things in humans. Gene therapy for rare diseases. And this is really cool stuff. The problem with rare disease, we say rare monogenic diseases. Genetic diseases caused by a single base change in the DNA. One letter can make a mistake and cause this disease. Out of that 3.6 billion bases, one in the wrong place can cause a problem. Diseases like cystic fibrosis, for instance. What scientists are doing now, and it's happening in many places and many companies, is they're going back and taking the virus that used to be something we were afraid of, disarming the virus by taking out the genes that made it infectious and problematic, and then putting in the genes that will correct the problem. And what's so cool about this is that this entire suite of viruses is now being used to reprogram specific cell types and allow people with rare monogenic diseases, that some which were fatal, to live completely normal lives. A good example of this is the adenovirus, which you can stick in a new gene and now be able to use this virus to infect specific cells in somebody who's suffering from a problem. We've seen corrections of diseases of the eye, where a specific gene was missing and now has been corrected by the virus to give people sight for the first time in their lives. This is reality. It's happening already. There's other applications that are happening either by treatment of the body with the virus that now imparts that new gene into specific cell types, or you can take cells out of the body, transform them, or add the gene outside of the body and then put them back in. And this works great with blood cells. What's so cool about stem cells is that you can use something called the lentivirus, give it new instructions to correct the potential blood disease, and then install those back into the body just by infusing them back in. They make their way into the bone marrow, and then from that point forward, will make the correct copy of the gene the correct copy of the protein and solve the disease problem. This is going on now, mostly in clinical trials. The animal models look awesome. And while it's not necessarily happening in too many people yet, I'll show you one, it definitely is the new edge of technology in correcting problems in cells and then adding them back to people to solve an important genetic problem. One good example is something called Fanconi's anemia, something you've probably never heard of because it's a rare genetic disease. There's no big pharmaceutical company working on this. There's not enough money in it. But a small startup would certainly benefit from being able to solve a couple rare diseases using this relatively affordable virus delivery system. So this list on the side, which I know you can't necessarily read, covers many different, I just wanted to show you this just to give you an idea of the breadth of things that are being solved, from beta thalassemia through muscular dystrophy, through cystic fibrosis, lots of diseases, hemophilia, diseases that we know of, from Jerry Lewis's telethon to understanding them from hearing about bad cases of cystic fibrosis, which can limit somebody's life and be an awful experience for both the parents and the child. These look like they will be solvable through these therapeutics in the next couple of years. So what about biofortification? And this is a really cool technique to maybe be able to supplement food for the food insecure. One major problem is vitamin A deficiency, and we take this for granted in the industrialized world. But the amount of vitamin A on the head of a pin is what you need to have a normal immune system and normal vision. And this would last you for months, that one little tiny fleck of vitamin A. Yet when you don't get it, you lose night vision, as just at the beginning of the disorder. It later goes on to immune breakdown, diarrheal disease, and eventually death. And in the countries on these maps shown behind me, I get out of the way here, the ones as they get darker in color, they approach 90 to 100% of people living in vitamin A deficiency. So how can that happen? There's many different things that happen, from infertility, wound healing, delayed growth, all kinds of issues in vitamin A deficiency. But in the developing world, it frequently leads to immune system breakdown, diarrheal diseases, and death. It's a huge number of children that die every year from this simple deficiency of that fleck of stuff that we have in carrots. So where does vitamin A come from? It comes from carrots and leafy greens. What are the folks in vitamin A deficiency regions consuming? Mostly items that don't have much vitamin A. So whether you're talking about cassava, about potatoes, rice, matoke banana, which is like a plantain, corn or wheat, there's not a lot of vitamin A going around in world food staples, which are caloric bases of every single meal. So how do you get vitamin A in there? Well, carrots are loaded with it, as are many different plants, sweet potatoes, things like that. It's the orange stuff that we consume. And what if you could somehow take the genes that are in a carrot that control the biosynthesis of the compounds that lead to the precursors of vitamin A and install them in something like rice? And that's what's been done, that a gene from corn and a gene from bacteria were added to rice to make the rice kernels full of beta-carotene. This beta-carotene could then be administered in part of the normal diet, grown by farmers for a decent price, and be used to correct the problems where these diseases and disorders exist. And it's working. The Philippines has now approved this and is now going to be, farmers are growing it right now for this season. Sad part is, this was originally developed 23 years ago. So this has been a long time coming and a long time in the works, yet it's just now starting to reach the people who it was meant to serve. The best part is, the same technology has been added to other types of world food staples, potatoes, soybeans, enhanced levels in cassava, to try to get more vitamin A into these regions of the world. And biofortification is a very exciting thing going forward. How about a fast-growing salmon? Many of you heard about this in Canada because it originated here. The idea was salmon that are farmed are naturally being farmed in large pens and nets out in the ocean. The problem is they can escape, they have to use tons of antibiotics to control disease, and there's a lot of other issues with farming farmed salmon in this type of way. The other issue is natural salmon populations are not doing great, that they are suffering from overfishing in many different cases. So one solution might be, could you come up with a salmon that you could farm in inland tanks? But that's difficult because it takes many years to grow a salmon to a market size. So what if you can make it grow faster? The problem with salmon growth is that it's cyclical. It goes in spurts with the season. As the days get longer, the salmon grows faster. As they go shorter, the salmon grows slower. So it takes many years to grow a salmon. But what if you could turn on the genes associated with growth, full blast the whole time, and grow that salmon the market size in a much shorter time? Maybe be able to do it in 1.5 years rather than 3 years. These two salmon are exactly the same age. The difference is the one on the top has its growth genes turned on all the time rather than half the time. So this is a product of a company called Aqua Bounty. They grew their salmon originally on Prince Edward Island. They're now using it strictly to raise the eggs. But they are a company that now has been given clearance to be able to use this product and sell it mostly through food service in the States, I believe in Canada too. A lot of people have been opposed to the idea. They say that, well, what if the salmon gets into not natural populations? Is it safe? Well, it's been shown to be exactly the same as the other salmon. And we don't worry about it getting in with natural populations because it's raised in inland tanks in Indiana and Ohio in the US, so it's a long flop to the Atlantic. The animals are triploid. What that means is they have one set of chromosomes from mom, 2 sets from dad. That means they're sterile. They can't reproduce even if they did get out. So this is just multiple ways in which using barriers plus genetics to ensure that these do not have an impact in the environment. And chances are they wouldn't compete well against a natural product anyway. But this is a nice way in which biotechnology is helping to increase the affordability of something that many consider to be a luxury food, and it's salmon. Another thing we think about are just common products like toast and French fries. And you probably never knew that your French fries could be dangerous. I know poutine's a cool thing here. I like it. You know, skull and crossbones on that stuff, right? What's the deal here? So anytime you're frying a potato or something with wheat in it, sounded like I said weed, wheat. That's legal here, isn't it? I don't know. So wheat in it or coffee, you generate a finite amount of a compound called acrylamide. And acrylamide is an IARC class 1 carcinogen. It's there in vanishingly small amounts. So don't give up on eating your French fries. Enjoy. But it does add a certain amount of risk to something that we really enjoy. There's the scary looking chemical there. So are there ways to limit acrylamide production? Well, there's a couple of ways that it's been done using biotechnology. We understand that this comes about from the way in which an amino acid, a building block of protein, one of the molecules that's in all of our cells, but present in plant cells as well, is fused to sugar when it's heated. And it makes this stuff called acrylamide. The big trick is that it's a biochemical pathway. And on this side, asparagine is the compound that it starts with. And as that's heated, the asparagine, through the action of an enzyme called asparagine synthetase, is converted, you have asparagine that when it's heated, gets conjugated to sugar and creates this toxic compound. So if we could limit, let me just put in my toxic compound face there. Here's acrylamide at the end. So this stuff in the beginning goes through this reaction under heat to make acrylamide. So if we could limit the amount of this stuff in the beginning, maybe we could slow down or decrease the amount of acrylamide in the final product. You could have twice as many fries. Yes. So how do we do that? So here's that map again. And here's the stuff in the beginning, asparagine. So what if we could limit the production of asparagine? That's it here. And the enzyme that makes it is something called asparagine synthetase. The asparagine synthetase takes its precursors and makes asparagine. So what if we could get rid of this? Take that book off the shelf. And that's what scientists have done, is they've managed to eliminate this from potatoes and now wheat to still have products that are 100% the same, just missing this precursor that turns into a toxic compound upon heating. Pretty cool stuff. Just by eliminating that, you eliminate that compound, and you can enjoy your toast. Curing childhood leukemia. This is another huge breakthrough for biotechnology by taking books off the shelf. And this is really cool stuff. Childhood leukemia used to be a death sentence for many kids because you can't give a bone marrow transplant especially easily, especially from non-like cells. They create what's called cytokine storm, a rejection process that is very dangerous, sometimes fatal to the recipient. And it happens because the cells have little proteins on the outside. They're decorated with these little signatures that say, "This is my cell." Scientists could take the cells from a donor and strip off all of those signatures, and do this by eliminating the genes that encode for those surface proteins. Then take those, now that you have a generic cell type, you can infuse this back into the child who's suffering from the problem. You ablate their bone marrow with radiation or chemotherapy, eliminate all their bone marrow, then put in these new stem cells that now have no identity as non-self. The body won't recognize them. And now these children are growing up with normal bone marrow that works just fine, that's not being rejected, and curing the leukemia. This is something that wasn't considered to be possible too long ago. You'd be in a permanent remission state, where now these appear to be cured. Another huge breakthrough in the area of childhood cancers and even more advanced age cancers is this idea that we can now reprogram an immune cell to go directly to the cancer cell and destroy it. This is cool stuff called CAR T cell therapy. It's a kind of immunotherapy. This 13-year-old, she's been completely cured, again, of a type of leukemia. It is an extremely aggressive type of leukemia. The reason it works is because cancer cells are normal cells that take on some strange identity. And when they take on this strange identity and start dividing in ways that they're not supposed to, they also accumulate specific signatures on their surfaces. And those signatures say, "I'm a cancer cell." Now you can take a white blood cell, a normal blood cell that's doing immune surveillance within our bodies, looking for problems so that it can go and destroy them. And you can program that cell with a match for that surface antigen on the cancer cell. Like if it's the lock on the cell, you now give this killer cell the key. And now this new engineered white blood cell, we've added a book to the shelf. That new engineered cell can go into the body and identify where the cancer cells are and destroy them. Like a guided missile. It identifies where those cells are and destroys them. It's been used successfully in glioblastoma, one of the most aggressive and traumatic cancers. It's being used in a whole variety of them. It works great sometimes, sometimes not so much. We're in the thick of the learning curve on this technology. But this kind of immunotherapy is something that we will see very commonly in the future. The next thing I'd like to talk about is, really, this is kind of the last one we'll talk about, is correction of a painful, debilitating disease that primarily affects folks of African origin. Sickle cell disease is a lifelong painful disease that happens way too frequently. Something like 100,000 people in North America are expressing sickle cell disease. This is Victoria Gray. Victoria Gray is like Neil Armstrong. She's like an astronaut. She's like the first man on the moon. She is a pioneer because she is the first person to be cured of sickle cell disease. And how did they do it? Sickle cell disease comes because of a single base error, a single mistake in DNA. The idea is that the single base change, this error, protected people on the African continent from malaria. And so you were more successful there when you had this mutation at times. But in modern times, this change in DNA creates a sickle-shaped cell, a cell that's not the right shape. And it causes all kinds of problems with the circulatory system, all kinds of pain in the joints. It causes a level of anemia. There are all kinds of complications depending upon the severity of the disease that make life absolutely miserable for people who have sickle cell disease. So what if we could correct that error? What if we could take bone marrow cells, go into those bone marrow cells, and use that technology called gene editing? We didn't talk about what they were called, but gene editing, going into the book and tweaking a letter and correct that letter, and then put those cells back into her. So take Miss Gray's cells, fix them, and put them back in. And then they go back into her bones, they proliferate, and now produce the correct proteins to make the correct cells. It's done by first pulling out her cells and fixing those stem cells. That now once they have the correct information, can go back in and make the correct blood cells that have the correct shape, not the sickle shape. And it works. Another way that this has been done is by using that method to correct or to delete just a couple letters. This is a complicated slide. But the protein that's the problem in the sickle cell is something called hemoglobin. It's the molecule that carries oxygen inside our blood. You make two kinds of hemoglobin in your lifetime. As a fetus, you make a type of hemoglobin that for some reasons we don't necessarily understand. Those genes turn off right after you're born. And a different kind of hemoglobin is made after. So in Victoria Gray, it's the ones that are produced later in life that are defective, that her fetal hemoglobin was just fine. So what if you could turn on this gene that has been turned off by making a little edit that normally was shutting off that gene and turn that gene back on? That's what's being shown here. Here's the mature type of hemoglobin, and here's the fetal type. We were able to turn the fetal type back on in her adult cells. So now she's expressing 100% fetal hemoglobin, and she's completely fine and over the disease. It's pretty exciting stuff. This is actually not necessarily new. This has gone on for a few years now. And Victoria Gray is an optimistic example of what we can do with this powerful technology. It makes me so happy to think about her because she's a really wonderful mom. And she had a debilitating disease that left her in constant pain, and she probably wouldn't have lived very long. She's now been functionally cured of this disease because of the power of biotechnology. Just a few examples. We could do a lot of things that I never even talked about, from therapies for solving problems in animals, extending lives of dogs, which would be super cool. They go too soon. Problems on the farm with pigs and cattle, all kinds of other, avian influenza, where we had to bulldoze 45 million birds in the last year that got sick from avian influenza and watch egg prices skyrocket. There are solutions for this in biotechnology that came out in 2011 that just haven't been implemented. So one title for this talk might be, "This is why we can't have nice things." That because of regulatory issues and other fear, that we can't necessarily deploy these things as fast as we would like to. And that's why I'm so glad you're here tonight. And I hope that I've inspired you to think about this in a little different way by looking at some of the examples that we can do very good things for biotechnology that we all care about. So what does the future look like? And this is the part that really blows me away, because I see how these tools, that the ability to change a single letter in DNA is only a few years old. I mean, we weren't doing this in 2015. We really started doing it in probably about 2018. It's brand new technology, and it's in the hands of everybody now. It's democratized science. You don't have to be a big company to do cool genetic DNA stuff anymore. You can be a small startup that can use this technology to solve an important problem, like Fanconi's anemia, or one of these rare genetic disorders. It has democratized the science. It allows small companies and other countries to be able to come up with solutions that are important for their people. Super exciting time. In the future, it's only going to go faster. The United States has made some investments in this area, and I think it's only going to increase. Of course, there are always people who will push back against it. And I hope that you're willing to talk to them in your conversations, especially in social media, and help them understand that everything, maybe that new technology always is looked at with some skepticism, and that's a very healthy thing, but that we shouldn't limit new technology because we don't understand it. and encourage them to get excited about thinking about these things, because they really are interesting technologies that really do satisfy the things that we all care about. The one big question for me is access. How are these newest medical breakthroughs going to be available to anybody who needs them? And how much will they cost? You know, will the 100,000 people suffering from sickle cell disease be able to get a $5 million therapy? Or will this be like a Jiffy Lube where you walk in and get a simple outpatient procedure of re-engineered cells? These are the big questions that we'll have to confront. Will there be regulatory hurdles that will be insurmountable? That's the other real big concern, especially these small companies with really cool novel approaches. Will there be enough money for research so that folks like me that work in the public sector can participate in these innovations and help drive the next wave of science? The bottom line is that science is taking off at an exponential rate. Discovery is going great. Innovation goes constantly, and it's only getting faster with better tools. The big problem is that even with life-saving technologies like the HPV vaccine, there are still places online where you can learn how this is killing you and your family. It's so important that we really are taking an active role in the communication of what these technologies are and what they aren't, and understanding the best sources of information, and directing our friends and family to them, having those sometimes awkward conversations across the dinner table or across the country using Twitter or Facebook. It's important to be participating in what these technologies are and what they aren't. And I hope I illuminated that for you a little bit today, just by showing you a couple of really important examples. So in conclusion, you know, when I was a kid, I'd always think about what are the most important things. And it was really, how are we going to continue to keep farming profitable? How do we have something for the developing world? How do we help the industrialized world consumer have good access to high quality food? And do this all with environmental sustainability. And that's stuff I've always worried about, still do to this day. It's so important that we think about the way technology can help us solve these important questions, because going forward, we're going to be relying on the best technologies to solve these problems as we going forward. It's really important that we understand what they are and what they aren't, and share that information with the people we care about. So I'll stop there, and please reach out if I can ever be of assistance. I would love to be able to talk to you more in the future about these technologies, or have you guest host a podcast. I would love to have that happen. Thank you so much. I'll be very happy to take your questions.